Thank you to the organizers for the kind invitation to present today. Today, we will discuss the importance of targeting adolescents, highlight some of the challenges that have been noted with oral PrEP use in adolescents, highlight what adolescents want in a prevention product, and then review clinical trials of long-acting antiretrovirals for prevention. Approximately 170,000 new infections occur in adolescents aged 10 to 19, primarily in sub-Saharan Africa. In sub-Saharan Africa, 6 in 7 new HIV infections among adolescents aged 15 to 19 are among girls. Young women aged 15 to 24 are twice as likely to be living with HIV than their male counterparts. In good news, the number of new HIV infections in this age group has steadily declined in the last decade. However, the rate of decrease in infections is not sufficient to meet targets for epidemic control, and programming gaps for adolescents and young people are greater than other population groups. Hence, adolescents are a priority population. TDF-FTC pre-exposure prophylaxis was approved by the FDA for adults in 2012, but not for adolescents until 2019. Therefore, research into how to best implement PrEP in the adolescent population has been ongoing. I will highlight the HPTN-082 study that recently published in PLOS Medicine. This study enrolled adolescents and young sexually active women aged 16 to 25 who were interested in PrEP and had a voice score greater than equal to 5, suggesting they were at risk at a level consistent with PrEP eligibility. They were randomized to the standard procedures, which included counseling, SMS text messaging, and adherence clubs, or they received this package plus information on their drug level from their previous visit, and messages are outlined as in the slide. There was a high uptake of PrEP, but there was no difference in the arms in PrEP continuation as seen in these largely overlapping lines from the time to discontinuation over 1 year. Also, there was no difference in the proportion of participants with tenofovir diphosphate levels greater than the threshold for good adherence, with only just 20% reaching this target. This project highlights several challenges for PrEP use in young women, and particularly the challenges with long-term adherence to a daily tablet. Even in a strongly supportive environment, the adherence requirements may be too much for this population. So long-acting products may be a solution. This is a study that asked adolescents and young adults what were their preferences for prevention products, and they were given specific options for various characteristics, specifically an implant versus an injection, the duration of time between doses, where it was available, the degree of soreness that might be experienced, and the location of an injection. Among the 807 young adults from Cape Town, including all groups, MSM, MSW, and AGYW, they stated that the duration between doses was the most important, with 1 year being preferable to 6 months to being preferable to 2 months. Arms were preferred, and women had a preference for a clinic over a pharmacy. The goal of decreasing the frequency of dosing is to the longest possible duration is clearly the area that we most need to target based on this research. So let's talk about long-acting antiretroviral HIV prevention. Injectable PrEP has several advantages, in addition to being a preferred stated method by populations that want to use a PrEP product. Every 2-month injections can directly tackle adherence issue, and it's very discreet. But it can't be removed once injected, so there could be potential for prolonged side effects, and there's a long pharmacologic tail after the last injection, which may have resistance implications in the event of breakthrough infections. Also, current testing algorithms may not perform as well in diagnosing breakthrough HIV infections with long-acting products. I will discuss HPTN 083 and 084 trials. These have recently reported their results. They have a similar design for different populations. The goal of these studies was to evaluate the safety and efficacy of cabotegravir in preventing the sexual acquisition of HIV in comparison to daily oral TDF-FTC. These were phase 3 multi-center randomized trials, double-blinded, that compared injectable cabotegravir to daily oral Truvada. These were the first studies to use an active comparator in both arms. Both studies had an oral lead-in phase to evaluate tolerance and safety prior to injection, and then entered the injection phase. Injection phase were every 8 weeks after an initial 4-week load. Cabotegravir arm participants received active cabotegravir and Truvada placebo, and those on the TDF-FTC arm received active TDF-FTC and cabotegravir placebo. After injections, an open label TDF-FTC phase was conducted. Both studies targeted and enrolled groups deemed at high risk of HIV based on local epidemiology. Specific risks are highlighted on this slide. In 084, sexually active women aged 18 and older living in sub-Saharan Africa were enrolled. 084 also used the modified risk score similar to the 082 study presented earlier. Neither study targeted in adolescents, but those under over 57% of the participants were 25 or younger for 084. The blinded phase of both studies were stopped prematurely by the DSMB due to evidence of efficacy. For 084, 40 confirmed infections occurred over nearly 4,000 person years of follow-up. The overall incidence of 1% suggested both products were highly effective. 4 infections occurred in the cabotegravir arm, equivalent to an incidence of 0.2%, and 36 infections occurred in the TDF-FTC arm, equivalent to an incidence of 1.86%. In reviewing the cumulative HIV incidence over weeks since enrollment in the intent to treat population, we see that women in the cabotegravir group had an 89% lower risk of HIV infection compared to the TDF-FTC arm. In 083, the results are similar. In the most recent analysis, a total of 52 infections were noted, 13 in the cabotegravir arm and 39 in the TDF-FTC arm. The incidence in cabotegravir was 0.41, and the rate in the TDF-FTC was 1.22. This corresponds to a hazard ratio of 0.34, favoring cabotegravir and resulting in a 66% reduction of HIV acquisition, favoring cabotegravir and meeting the definition for superiority. Cabotegravir was superior to daily oral TDF-FTC in preventing HIV, 89% in 084 and 66% in 083, and the injections every 8 weeks likely provided an adherence advantage over daily oral TDF-FTC. Additional studies are being conducted to evaluate safety, tolerability, and acceptability of cabotegravir for prevention among adolescents. Each of these open label studies will enroll 50 participants from selected sites that conducted 083 and 084. The goal of this study is to allow concurrent licensure or nearly concurrent of cabotegravir for adolescents. 2 large studies were conducted to evaluate the efficacy of the pivarine ring. In both studies, the product was found safe and the efficacy was very similar, 27% in Aspire and 30.7% in the Ring study. Younger women, though, aged 18 to 21, had no protective benefit in either study. 5 open label extension studies have been conducted on the Ring. Dream and Hope were open label extensions of the original participants. REACH is an important study evaluating young people's attitudes towards the ring. DELIVER focuses on pregnancy, and Be Protected is evaluating the ring in breastfeeding women. For the REACH study, it has completed enrollment, and the study is expected to report its findings in October 2021. In summary, let's review the prevention products I've mentioned. Oral PrEP has a wide efficacy, largely dependent on adherence. The Deprivaring ring has 27 to 31% efficacy, but it varies by age and adherence to the product. An injectable cabotegravir every 8 weeks has 89% reduction in HIV acquisition compared to daily oral TDF-FTC. Oral PrEP in the ring can easily be stopped or removed, and the ring has less possibility of HIV resistance due to its lack of systemic absorption. Oral PrEP is used in adolescents, and results are pending for Deprivaring ring and cabotegravir. What's on the horizon? While we anticipate the addition of injectable cabotegravir to the prevention toolbox, we know these options still may not be enough to fit everyone's lifestyle, particularly complicated adolescent lives. Islatravir is the next ART prevention strategy for consideration. It is a nucleoside reverse transcriptase translocation inhibitor. It will be evaluated in a randomized controlled trial to begin in 2021. It will compare monthly oral islatravir to daily TDF-FTC. Notably, the same product can be formulated into an implant that may allow another long-acting option. This islatravir implant provided drug concentrations above the targeted PK threshold for at least 1 year. Phase 2 studies are planned. And the newest agent on the HIV prevention block is lenacapavir. This is a capsid inhibitor and has a novel mechanism of action. This ART has been most studied in treatment-experienced patients, but may be well suited to long-acting prevention. It can be given subcutaneously every 6 months. It has been announced that lenacapavir will be added to a clinical trial that is evaluating TAF-FTC and TDF-FTC in women. And as we evaluate long-acting ART for prevention, we need to recognize that multi-purpose prevention will also be moving forward, that will combine contraception, HIV prevention, and STI prevention. Cross-cutting prevention, along with integration of sexual health services and care, will benefit the complex need of adolescents. Long-acting injectable antiretroviral with cabotegravir has been shown to be safe and effective. The Deprivaring ring has recently received WHO endorsement. Adolescent studies for both products are forthcoming, and we expect the products to be available in the near future. Additional long-acting products are entering clinical trials, and fortunately, adolescents are going to be included earlier in the research process. All new products will require implementation science research to translate evidence to action and impact. I'd like to acknowledge the support from the funders and others in the studies that I have presented. Thank you very much.